hypothesis testing. Previously, Hulin talked about statistical inference by point estimation and also interval estimation. Today, I'm going to talk about statistical inference by hypothesis testing. After that, I'm also going to briefly mention about the data problems and how should we deal with those problems. For hypothesis testing, we need to understand what is the null hypothesis, the alternative hypothesis, also the type 1, type 2 error, sample size, test statistics, and the p-value. After I talk about hypothesis testing, I will mention about data problems and also some very complicated data and how to deal with them. Before we talk about hypothesis testing, we need to understand what is the statistical inference. Statistical inference is systems of procedures that can be used to draw conclusions about a population through a randomly selected sample from the population. Here, the plot gives us the idea. Here is a large population. We cannot get the information for every one of the population, right? So we randomly draw a sample from this population, and then we do collect the data and do some statistics. We get to understand the samples, because the samples is randomly selected from the population, so that we can draw conclusions about the population from this sample. Here is the example. What's this example I'm going to use later on to explain the idea. It's the H5N1 vaccine trial. It's an open-label phase 1 evaluation of single-dose vaccination H5N1 vaccine in subjects who were previously vaccinated with a vaccine made from antigenic distinct variant of H5N1. So here we define the population. The population is a subject who were previously vaccinated. So it's not everyone. So this is our population. The purpose is to determine whether the immunization with antigenic variant could prime for response to a single dose of current vaccine. So here, our population is the number of the eligible subjects. That's the subjects who were previously vaccinated with antigenic variant vaccine of H5N1. The total number of subjects is 117. So this is our population. Our population consists of 117 subjects. The number of participant subjects, this is just we have the study and we send notice or any format to the eligible subjects, and some subjects come. In total, we get 28 subjects. So this 28 subjects is our sample. Our 28 subjects out of population of 117. So this is what does it mean by population and also by randomly selected sample. Now we are going to talk about hypothesis testing. Hypothesis testing is a statistical method of making decisions using data, whether the data is from controlled experiment or from observational study. It doesn't matter. So we need to understand the null hypothesis, alternative hypothesis, also the type 1, type 2 error, sample size, test statistics, and the p-value. I'm going to talk about them one by one. The null and the alternative hypothesis. In general, the null hypothesis is typically the default position. That means low difference or low effect between low difference of between two groups or low effect of interesting treatment. The alternative hypothesis is typically the opposite. That means there is difference between two groups or multiple groups, or there is the effect of treatment. Before we do experiment, we do hypothesis test, we need to tell two things, we need to tell them apart. One is about choose, another is a decision. These two are different. in choose, because we need to understand what is the type 1 error, what is the type 2 error, so we need to tell them apart. If in choose, the null hypothesis of low difference is true, but we make a decision that is the null hypothesis is not true, we accept the alternative hypothesis. In this case, we made a type 1 error, that's false positive. But if in truth, the null hypothesis is false, so, well, in other words, the alternative hypothesis is true, but we accept the null hypothesis. In this way, we make a type 2 error. In general, we want to control the false positive rate because we think normally people think it is more serious. So normally we control the type 1 error with the false positive rate to be 0.01 or 0.05. We usually use alpha to denote the type 1 error and beta to denote the type 2 error. Another associated words with type 1 and type 2 error is the significance level. So, for example, we set the type 1 error to be less or equal to 0.01. So the significance level will be 0.01 or 0.05. Another word for type 2 error is the power. Power is when the alternative hypothesis is true. That's in this case, or the null hypothesis is false. And we accept the alternative hypothesis, or we reject the null hypothesis. So in this case, here is the probability of or possibility of reject the null hypothesis, accept the alternative hypothesis when the null hypothesis is false. This is the power. Here the graph gives us some what is the significance level and how to decide the criterion rejection region based on the significance level. So it's if we see the significance level is 0.05, so from here and up and worth of the star lower, it will be a rejection region. As I previously mentioned, the power is 1 minus the type 2 error. Another thing good to know is the type 2 error increase when the type 1 error decrease, so they are associated. Now we come to sample size. Here I plot stop working. Here you can see I plot different scenario for different mean difference between two groups. The interest is compare two groups in some continuous variable, and I want to know whether there are difference in the continuous variable between these two groups. So I select two scenarios for the mean difference. One is 3, one is 4, and also I select a different standard deviation. One is 8, one is 9. So you can see the different color is denote different standard deviation. The circle and the plus sign denote different mean difference. You can see as the mean difference so is for same color but a different symbol. The plus sign is for when mean difference is 4. You can see when the mean difference between two groups is bigger, but we require smaller sample size. Here is total sample size required. Compare these two red curve. However, if we compare these two lines, different color but all with the circle symbol, you can see when the standard deviation, the blue line, is bigger, we require larger sample size. So in this way, we know the sample size is inversely correlated with the group difference and positively correlated with the standard deviation. So the sample size depends on the significance level. and also depends on the power, depends on the clinical difference, and also other population parameters like standard deviation. The question is, before we do experiment, where I can get this information? One way to get it is to do pilot study. If we don't have, we cannot get the information from literature, we do pilot study, very small study, to get some idea about the mean difference and also the standard deviation. Another way is to, from the literature, people published a similar experiment or similar study, so we can borrow the information from literature. If we don't have resources to do pilot study, and also we cannot find any information from the literature, another way to do that is we use percentage change and also the coefficient of variation. Because as I mentioned previously, the sample size is positive, is negatively correlated with the standard, the interest difference, right? Group difference. And also is positively correlated with the standard deviation, if we say delta, sigma. So you can, and also we did add other items. So in this way, if we use correlation of variation equal to the standard deviation divide the mean. So that means the correlation of variation will be, from here we can get the standard deviation equal to mu times the correlation coefficient. And also we express the interest difference as the percentage change of the mean. So for example, we see 8% times the mean. So in this way, the sample size will be equal to mu times CV divide the data t, so data d. So it was 8 times mu, and also add other items. So you can see the mean can be cancelled out in this way. This is when we cannot get any information about the mean and also about the standard deviation, we can use this way. And also, even we use this way, we need to have some knowledge about how much the percentage change will be interesting, clinically interesting. We need to have this knowledge, and also, of course, based on our resources. Here is the plot. Here we want to compare two groups on the mean plasma peak concentration between two groups. So suppose we don't know any information about this, the mean difference and also the standard deviation. We can use this way to do that. So we see, if we say we want to know, here is mean difference is 30% change or 40% change. And also we can suppose the standard deviation, the correlation coefficient is like 30, is 0.3 or 0.4, 0.6, 0.5, any number. And then we can get beautiful plots. From the plot, we can decide, for example, we see here, if we have for the red line, and here, of course, the before experiment, any experiment, we select the type 1 error and the power. Normally, type 1 error is 0.05 and the power be 80%. So for here, if we see the red line, to have 80% power, the red line is for 0.3 correlation coefficient. For 40% change, the total sample size required will be around 20. So we will decide whether 20 will have the resources, or we have more resources than that, and then we can go to other place. So by giving these plots, you can select how much one is, what kind of statistical clinical difference will be interesting to you or interesting to the in general. And also we need to have some idea how much the variation will be using the correlation coefficient and how much resources we have. So we can do beautiful plots and then put all the information together, select a sample size include take account of all of these informations. It's very easy to do for this kind of plots and also sample size calculation by R, by SAS, and also I believe by other software. Now it comes to test statistics. Test statistic is a numerical summary of a set of data. When we choose test statistics, most important thing is to know the sampling distribution of a test statistic under the law hypothesis can be determined so that we can get the p-value. And also, under the given null hypothesis, a test statistics have to satisfy the following criteria. That means one meet the following criteria. That means a value of t close to t0 present the strongest evidence in favor of the null hypothesis. If it is far from t0, it will against the null hypothesis or in support of the alternative hypothesis. Once the test statistics is decided, we know the null distribution, the sampling distribution of the test statistic under the null hypothesis, and then we can get the p-value. Is the probability of obtaining a test statistic more extreme than the actual sample value given the null hypothesis is true. With this information, we compare the p-value with the significance level. We can make a decision whether we reject the null hypothesis. And what is equal to low under the null hypothesis, the p-value is uniform distributed. Here is an example I briefly mentioned previously. I'm going to use this example to show you how we can do hypothesis tests like t-test and f-test, chi-square test. As I mentioned, this is for H5N1 vaccine trial study. The information we have, data we collect is, we have PBMC collected at day 0, 7, 28, and 56. And also we have the clinical information or lab information, antigenic B cell, total IgG, and H5VN specific cell count, and also HLI titers. For clinical information, we have age information, sex, and also we have prior seasonal flu vaccination status, whether a subject get prior seasonal flu vaccination or not. First, I'm going to talk about the t-test. If we want to test whether the antigen-specific cells, the total IgG, are the same between the group with prior seasonal flu vaccination or not. So we are going to compare two groups. We want to know whether at each time point there is a difference in total IgG. The null hypothesis will be the same. There are no difference between these two groups. The alternative hypothesis will think there is a difference between these two groups. The significance level will be 0.05. The power we know, normally we choose 80%. And also we need to calculate the test statistics. Because here we are testing mean. So when we compare mean, usually we use t-statistic. Later I'm going to check why should we use t-statistic instead of others. Once we decide the test statistic, we can obtain p-values. Here is the raw data. So you can see this is the total IgG. I didn't do any transformation. You can see at day 0, this is a box plot. You can see we have some value, there are some variability within the data. Some, like here, looks like an outlier. And also the variability between these two groups quite counts for example, like day 0, they look quite different. So from the plot, we can see looks like there are difference at a baseline between these two groups, and possibly at day 7, but not at day 28 and day 56.
What's on your y-axis?
Y is the IgG, is the cell count. So the first thing to compare two groups, what we can do is use Lamprometric test. Here I use Wilcoxon rank sum test at each time point. So we see at day 0 there are no significant difference between these two groups, but there is a significant difference between these two groups at day 7. The good thing about the Wilcoxon, the Lamprometric test is simple and convenient to use, but the drawback is not so powerful. Go ahead.
In the comparison of means between groups, when is ideal to use the T and when the Wilkinson ransom?
Yeah, I will show you later. I will show you. And then the point is we need to check the normality. If we can assume the sampling of, because to use the t-test, the one sampling is the data have to normal, like a normal distribution, right? So we need to check the normality. The top 4 plots are the raw data at each time point. You can see it's quite skewed to a depth. So it doesn't look like normal distribution, right? But however, if we do some transformation, like log transformation, this is the bottom 4 plots. They are quite good, right? Very look like normal distribution. So in this case, we can do log transformation use a t-test. Why? Because it's quite look like a normal distribution and it is more powerful. So here is how we calculate the t-statistic. I'm not going to talk about it in detail about this formula because nowadays it's very convenient to use software. You can do it, yeah. So, but still we need to understand the idea and the sampling and how to do that. If we understand it, and then we can use software to do it. Otherwise, it's hard to explain. Here is how t distribution look like. So you can see the t distribution for the blue line is t distribution with degree of freedom 1, and then the red line is degree of freedom at 3. The green line and also the yellow line are degree of freedom at increased. So you can see when the degree of freedom increase, the t distribution is very close. The black line is the standard normal distribution. So are very closer to the normal distribution. And also you can see for our example, because the t distribution, the degree of freedom is 18, is very close to the normal distribution, and also, of course, so as a reason, the critical value is very close to the critical value and the normal distribution. Here is the test result by t-test. As I mentioned, we use the log transformation because after log transformation, the data look like normal distribution. So in this way, if we use t-test, it's more powerful. Here is the result. You can see now the scale of the Y scale is very small, not like before, it's like 10,000, several 10,000s. And also you can see here the group difference at the baseline, there is difference at the borderline significance. At day 7, they are highly significant from each other, the two groups. But for day 28 and day 56, there are no significant difference. Here is the test result in table, and I show you what's the observed difference at each time point between these two groups. What's the t-statistics? You can use the formula I presented before to calculate by hand. Of course, you can do it by software. And what's the confidence interval? And but if you use R, and later on, Hua is going to talk about how to use R to do statistical analysis. It's very powerful, and also it's free. It's very convenient. Yeah. If you do it by R, it's very easy to do. It's just simple. One statement, you can get the result. This is p-value. Here, you have to know, we assume equal variance between these two groups, because after log transformation, looks like they are quite similar. Here, because we do tests at 4 time points, we need to do multiple test adjustment. Why? If the null hypothesis is no difference in total IgG between two groups at each time point, the alternative hypothesis says there is group difference at least at 1 time point. So, because for each test, we assume each between group comparison was made at the alpha significance level, then the probability of finding at least one significance will be greater than alpha. So we said before, we want to control the false positive rate. So we don't want this to happen, to have the type 1 ISO greater than alpha. So we need to do the multiple test adjustment. I'm not going to talk about in detail how to do that, but I do want to talk about the most convenient way, is the Buffer-Rowley correction. By Buffer-Rowley correction, it's very easy. So originally, if we do one test, the significance level will be alpha. If we do n tests, the significance level will be changed from alpha to alpha divided n for n comparisons. Or in another way, the p-value can be get by this formula, is the minimum n times p and 1. The advantage of Buffer-Rowley correction is simple and easy to use, but the disadvantage is in some cases is too conservative, and also it's reducing power. Well, of course, there are other good ways to handle that. For example, like the false discovery rate used a lot for gene microarray data analysis. In this hour long, Rui Hu is going to give more information about the multiple test adjustment, also about this method. So I'm not going to talk about them in detail. Here is the test result if we apply the multiple test adjustment. The first several lines are the same as before. The last line is adjusted p-value by Buffer-Rowley. You can see if we use the formula, is a minimum of, because we have 4 tests, 4 time points, we have 4 tests. So it will be the minimum of 4 times the p-value or 1. So if we applied the multiple test adjustment, the only significance will be at day 7. So there is a significant group difference at day 7. Next one, I'm going to talk about F-test. The assumption will be same. We want to test whether there is significant difference in total IgG between the group with prior seasonal flu vaccination and the group without it. Here I just use the same number because I don't want to give you many different examples to, we do not have so much time. I just use the same example. But in general, for F-test, we use it to do multiple group comparison. So the null hypothesis will be same. We assume there are no difference between these two groups, And the alternative hypothesis says there is a group difference. The significant level will be same. In this time, we will use F statistic by ANOVA. Why should we use F statistics? Actually, the F statistic is from one-way ANOVA. One-way, what does it mean by one-way ANOVA? That means we divide, we have the dependent variables outcome, right? How to explain the variability for the data? We divide we just divide the variability into several parts. For example, here the interesting, one interesting is the factor is the pre-flu vaccination, right? So we want to know whether this has an effect, can explain some of the variability of the data. So we can use the one-way ANOVA to do that. We can divide the variability in the data into the variability explained by the factor of pre-flu vaccination, and also another part of variability may be due to the measurement error or may be due to other reasons. The F-test can also used to one function of F-test one-way ANOVA is used to assess whether expected value of a quantitative variable within several predefined groups differ from each other. This I talked about just now. Another functionality of F-test, we can use it to compare statistical models in order to identify the model that best fits the population from which the data were collected or sampled. By ANOVA F-test, the advantage is when we have multiple groups, we can do by ANOVA F-test, we can simultaneously compare multiple groups to see whether there is a difference among multiple groups. But the disadvantage is if we reject the null hypothesis, we have local rule, which group has a bigger mean or small mean. And also we don't know the directions. We just know there is a difference among these groups, but we don't have a clue which is bigger, which is smaller. Here is how we formulate the F statistics. Is between group of variability divided within group of variability. It has a 2 degrees of freedom. For our sample, because we have just 2 groups, so the first one will be the first degree of freedom will be k minus 1, so 2 minus 1 is 1. For the second degree of freedom is n minus k, n is the total sample size, k is the number of groups. So in this case, we have 20 minus 2 is 18. It's easy to understand why we use this to do hypothesis test. Because when there is a group difference, the between group variability will be much bigger than the within group variability, right? So we will get a very big number. The big number will reject the null hypothesis. So more variability can be explained by the group difference, the between group difference. Here I also put how the T distribution correlated with the F distribution. So that means if the X is from T distribution with degree of freedom n minus k, then the X square will from the F distribution with the degree of freedom 1, the second degree of n minus k. And also, if we have 2 random variables, independent variables, x1, x2 from chi-square distribution with a different degree of freedom, by this way we can get the F distribution. Is the chi-square distribution of the first one divide the degree of freedom, and divide the second chi-square random variable divide its degree of freedom. We get an F distribution. Here is how F distribution look like. for different degree of freedom, because it has 2. When degree of freedom is equal to 1, the first degree of freedom, the second degree, even the second degree of freedom are different, 18 or 100. They quite look like similar. So in this case, the first degree of freedom dominates. But if the first degree we change to 100, in this case, the second degree freedom is 18 or 100. They don't look like each other. So they're not so skewed. Here is the test result by F Statistics. I put the F Statistics and what's the degree of freedom 118. The R code, how to do that? You can see it's very simple to do that by R. And the p-value. It's worth to mention that the p-value here is exactly same as the t-test. 0.07 is here. You see, it's exactly same. Why? Because just now I mentioned there is this relationship between F distribution and the T distribution. Do you have a question?
Yeah, I wanted to ask you, what would the F distribution look like, or if you know of any studies pre and post normalization? Because pre-normalization we may get false discovery, but after normalization.
You know what? For both F distribution and for F test and the T test, one assumption is the data should be look like a normal distribution. Is one assumption. Another assumption for one-way ANOVA is it assumes the standard deviation between these two groups are same. The third example is how to do chi-square test. If we want to test that the HAI titer response to vaccination is either we have the response or we don't have the response, is associated with the age. We divide it because the age range in our study population is between 40 to 58. So we divide it into two age groups, 40s or 50s. We want to know whether the HAI titer response status is associated with the age. So that is two categorical data. One is her HAI response status, yes or no, or the age categorized age, 40s or 50s. So here on the right side is the continuous table. Is how many people in 40s has response or do not have response. This is a continuous table. How to do the test? So we need to formulate the null hypothesis and also the alternative hypothesis. In this case, the null hypothesis will be the HAI titer response to vaccination is not associated with age. The alternative hypothesis will be yes, they are associated with each other. Still, we select the same significance level, but in this case, we use chi-square test because we are testing the relationship between two categorical data. In this, we should use chi-square test. By chi-square test, we can use it to do goodness of fit test of observed frequency distribution compared with the theoretical distribution. For example, we have a frequency. We want to know the data is from Poisson distribution. So we have observed count. If we, under the null hypothesis, we think it's from Poisson distribution, we can get the expected value under Poisson distribution. And then we can use some formula to get the test of chi-square. You can calculate, and then we can decide whether the data is from Poisson distribution or not. This is one functionality of chi-square test. Another way is we can use chi-square test to test the independence between categorical data. This is how chi-square distribution look like with different degree of freedom, 1 to line. So the yellow line is for degree of freedom 1, And then when the degree of freedom increases, the shape changed. Here is normally when we do chi-square test, we need to know the contingency table. Here the table, because we are doing the test to the association between two categorical variables, we need to find the for each cell what's the frequency, and also the though means the sum of those, and also sum of columns. Here is how we calculate chi-square statistics by hand. This is a formula. Under the null hypothesis, what's the expected value of cell frequency? And then after we get the expected value, we can formulate the chi-square statistic by this formula. Is the observed minus the expected squared divided the expected value. Is in this case, if we assume there are rows, there are C columns, the degree of freedom will be R minus 1 times C minus 1. Here is a real example. As we mentioned just now, for HAI response status, yes or no, age 40s or 50s. So this is the contingency table. Here we can use the formula to calculate chi-square statistics and then get the p-values. But one problem here is the cell frequency is very low, 2. Normally we use 5 as a cut point. If the only cell frequency is less than 5, we need to do some format of adjustment, or we need to use non-parametric weights. Because in this case, when cell frequency is very low, the test statistic does not approximate the chi-square distribution. That's the reason we need to do some adjustment. One way to do that is Yada's correction for continuity. Another way is the non-parametric test, is the Fisher's exact test. Also, we can get a p-value by Monte Carlo simulation. Here is how we use each of these adjustments. For Yada's correction, this is the formula. Previously we do not have the minus 0.5, but here we add this one, and in this way, it's the test figure will look like more like a chi-square distribution. If we can also use the Fisher's exact test. In this case, we assume the row and the column margins are fixed. Under the null hypothesis, there is no association between these two categorical variables. The N11, the cell at here is the N11. Is the cell frequency at here, will be hypergeometric distribution with these parameters, and then by this way we can calculate the chi-square, the p-value by Fisher exact test. Another way to do that is we can do resampling. We fixed the row and the column margins. We randomly select the samples, and each time for each resample we calculate chi-square statistics. If we do the process many times, we can get the sampling distribution of the chi-square statistic. In this way, we can calculate the p-value. Here's the test result. It shows if we do not do any correction, we just use chi-square statistics, the p-value will be 0.23. But if we use Yada's correction, the p-value will be 0.43. By Fisher exact test, the p-value will be 0.40. If we do resampling, the chi-square test will be, the p-value will be 0.41. So you can see the last three are very close, quite consistent, but not the first one. The reason is we have the cell frequency is very small. The conclusion is HI-title response to vaccination is not associated with the age group. Here is the relationship among common distributions, because I mentioned earlier about the relationship between T distribution and the F distribution, chi-square distribution and the F distribution. In fact, the theoretical distributions are correlated with each other. In some way, if you do some transformation, you can change to another distributions. For this project, I didn't do it by myself. You can Google online. And there even you can get a more detailed information how they correlated to each other. Here is the standard statistical testing methods. I put usually the common case. What should we do? Like if the type of data is binary, and we have just one samples, we want to compare. We will see the positive rate. We want to see whether it's positive is 0.3. In this case, we can do one sample binomial test. Or if we have two, we want to compare two groups, the positive rate, but the two groups are independent. In this case, we can do chi-square test or Fisher's exactly test. Or if we have more than two groups, what should we do to compare them? We still use chi-square test, but these all we assume the groups are independent. If the groups are not independent, they are paired or correlated with each other. In this case, what kind of test should we use? I put all of them for binary data and also for nominal data. What should we do? What kind of test to use? Whether they are correlated or they are independent.
Is it possible to use a combination of those statistical methods and wherein you can apply not a single method but to have a combinatorial methods?
Like a very simple example is I presented just now, like to compare the two group, right? Two group difference. I can either use the t-test, I can also use the f-test, right? So because the reason is the t-test is correlated with the f-test, you can. So in some cases you can use either way. The result, whatever you use, whichever method you use, you need to check whether the results, the conclusion are consistent. So if they are not consistent, you need to figure out what's the reason, whether the data met the assumption. Next, I'm going to talk about the data problems. One type of data problem is outliers. Another one is missing data, how to deal with missing data and also censored data. Here is how we define the outliers. We use a box plot. We get the first quarter, and also the quantile, the third quantile. The distance between first quantile and the third quantile we call the interquartile range. If for between the third quantile and the third quantile to plus 1.5 times interquartile range, here any value beyond this point we will call it outlier. Also any value less than here, the first quantile minus 1.5 times the interquartile range. Any value less than this point, we will call it what's called an outlier. Here's how we do it by if we use normal distribution, what's the percentage of interquartile range and also the range up beyond the what's the probability beyond the or being outlier. Here is another plot. You can see how to define the outliers.
So is this mainly applicable in the case of box plot?
This is from box plot. Yes. I put the plot here is I want to explain how you define the outliers by box plot. How to if you want to know what's the outlier, you need to know the first quantile, third quantile, and what is the interquartile range, and then you can define the outlier. working with outliers. How to deal with outliers? Easy way, very easy way is just exclude them. But it's more accurate when the underlying distribution is confidently low. For example, we know the data is from normal distribution, right? In this case, we could exclude that. Because another way is in regression analysis, we can decide, exclude influence points, which exhibit large degree of influence on parameter based on the measures such as Cook's distance. This also can be done by software. If you fit regression model, you can ask for the Cook's distance to detect those outliers, and then you can choose to exclude them. Also, we can use robust algorithms to those, for example, quantile regression, which quantile regression is robust to outliers. Another method is if we know the cause of outlier, we can incorporate the information into a model structure, because like mixed models. Another type of problem is missing data. We have different type of missing data mechanism. For example, missing complete at random. When the distribution of missingness doesn't depend on either covariate or outcome. What's missing at random? When the distribution of missingness may possibly depend on covariate but not on outcomes. The third type is missing not at random. The distribution of missing depends on both covariate and the outcome. So I'm not going to talk them in detail how to do that. And in this case, I think you need, if you have the missing problem, you need to consult with the statistician how to deal with them. For censored data, it occurs when the value of a measurement or observation is only partially known. We can use different statistical approach to deal with censoring problem. For example, severe analysis, the topic analysis. More complicated data, like longitudinal data, time to event data, time series data, and raw data was summarized data. Just give one example. Longitudinal data. Here is we want to test the H5VN specific cells counts over time. Here is the raw data plots. I also want to compare the cell counts whether the pre-flu vaccination has effect on the H5VN specific cell counts or response. Here is the raw data for different group for with pre-flu vaccination or group without it. You can see variability is very big, and from the data it's hard to find whether there is a significance, right, between this group. It's hard to find from the data. For how to answer the question, well, what should we do? There are several ways we can do that. The simple way is repeated use of t-test on difference from D0. Because now we want to know whether the pre-flu vaccination has effect on the response. So we don't want to do test just on the raw value. Let's do test on baseline, like IgG at day 7. We want to do test on difference. Because the difference in some way is like we take account of the cell count at the baseline, right? So we can do the test on the difference from D0. For example, t-test on the cell counts at day 7 from the difference from day 0. The drawback is we didn't take, in this way, we don't take account of data correlation over time. Because the data from the same subject are correlated with each other. Of course, it's inefficient because we just use partial data, do it one by one. And also we have inflated type 1 error. More complicated ways we can do that is repeated ANOVA, because those data from same subject are correlated. We collect the data from same subject for several times. This we can use repeated ANOVA, or we can use a big model, like a parametric model or non-parametric model. Here is the plot of difference. Go ahead.
Can you use a paired t-test?
practice, we want to compare the difference between two groups. Those two groups are independent, right? We do.
Between the time points in one group, that's what you said.
We want to know whether the pre-flu vaccination has the effect on the response, right? Yeah. So if we do paired t-test, you just compare the within group. If you do pair test, it's within group comparison. We want to do pair test between group. Yes. Here is the plot of difference. You can see we have negative values. If we use the t, so the plot of difference, and also you can see we have negative values and what kind of test we can use. One way, of course, as I mentioned earlier, is we can always use Lamprometric test. This plot of difference, we have negative values, and this is raw difference. T-test will run some tests. The difference is to loud like normal distribution. And also with quite a good percentage of negative values, it's hard to find a correct way to transform data. Normally, I said previously, we can use log transformation because the data doesn't look like normal distribution. And if all the differences are positive number, we can try log transformation, right, to see whether it's normal-like. But with many negative values, we cannot do that. We also don't want to just throw them away. So we can always, in this case, we can always use the Lamprometric test, but it's less powerful. Here is the p-value. If we use the Wilcoxon random sum test, the conclusion is pre-flu vaccination has low effect on day 7 on response, also at day 28, day 56. Now, how about we try complicated method like repeater ANOVA? Here is the raw data plots. You see it's quite skewed to the left, doesn't look like normal distribution. And then if we do log transformation, it's more like a normal distribution. And then we can use the repeater ANOVA. You can see this is the raw data and this is transformed data. The raw data, we cannot see anything. But for transformed data, you can see the slope between these two groups are quite different. After we do transformation, it's look like normal distribution, and also the slope is bigger for the group with pre-flu vaccination. Here is the model to do the repeated ANOVA. Yeah, I have to finish it soon. Here's the model. I'm not going to talk about it in detail. And here's the result by repeated ANOVA. You can see the first part is by Lamprometric test on the difference from baseline. The second part is repeated ANOVA on the raw data, but the raw data doesn't look like a normal distribution, right? The third part is repeated ANOVA on the log transformed data. You can see the conclusions are different. When we do repeated ANOVA on transformed data, the conclusion is there is significant, the pre-flu vaccination that has a significant effect at day 7 and also at day 56. But it has a borderline significance at day 7. The reason is by repeated ANOVA, we take account of correlation within the data within same subjects. The idea for statistical test is we try to take account of as much variability as possible. All the statistical test is against to unexplained variability. So by repeated ANOVA, we take account of the within subject variability, and also we transform the data to like it more like a normal distribution. It's more powerful. That's the reason the conclusions are different. Okay, the point is for all of the complicated data, you need to consult and involve professional statistician. Have any question?